Good morning, class. Welcome to Microbiology 101. I am your professor, Miss Mari Filer, and today we will be learning about a pathogen by the name of Neisseria meningitidis. So today we're going to start off with a focus activity. So based on what you're observing now or on previous knowledge, what can anyone tell me about this bacteria? I can describe that, Miss Filer. You. Well, based on the round shape of the pathogen, I think it could be coccus, and then it's also in pairs, so that would make it diplococcus. Also, in the gram stain, it showed up as a pinkish color, which means it didn't retain the crystal violet stain, which would make it also gram negative. Am I right? Great job, Haley. Did everyone hear her? As you can see, the bacteria is round shaped, so that makes it coccus, and since it's in pairs, that would make it diplococcus. Also, on the right hand side, you can see that as a gram stain, they stained pink, which means it did not retain the crystal violet stain, which would make the bacteria gram negative. Great answer, Haley. Yes, you were right. Next, we're going to talk about how we identified in meningitidis. So the first step in identifying was the polymerase chain reaction, also known as PCR. Now this is a technique used to make many copies of a DNA region. So we created a valve and we added a master mix, 2 primers, PCR water, and a template to create the PCR reaction. And after that, we submitted it for agarose gel electrophoresis. This is a technique used to help visualize the individual fragments. So we sent off our PCR results and we actually dropped our results of PCR into agarose gel, and later on that night we were given our results. Next, after obtaining a DNA sequence, we could submit it into a database for analysis, which brings us to DNA sequence analysis. In a few minutes, you will see the actual DNA sequence, and we copy and paste it into a database. And what pulls up is different results of what this pathogen might be. After that, we submit our DNA sequence into another database called BLAST, where it will go through the same thing, but it might not give us the same results because it's in a completely different database. And lastly, after obtaining the different analysis, we will create a phylogenetic tree, which will basically show the different genetic trees that our pathogen comes from. First, we have the results from the polymerase chain reaction and the agarose gel phoresis. This right here is our PCR and where we conducted our agarose gel electrophoresis. As you can see, ours is A3 and it kind of looks similar to the latter. However, that is not how it was supposed to turn out. It should have looked a little more similar to A7. Although we don't know exactly why our PCR and agarose gel electrophoresis turned out this way, it could have been a number of things. Maybe incorrect manipulation during PCR, or it could have been incorrect steps used during the agarose gel electrophoresis. However, that is not going to stop us on our journey to identifying our pathogen. Next, we obtain our DNA sequence. So each individual was given a unique sequence to identify. And as you all know, DNA contains adenine, guanine, cytosine, and thymine. And that's what all of these C, A, G, and T letters represent. This information is then submitted or entered into a database to help analyze and identify the pathogen that you might have. The results. So, like I just stated, our DNA sequence was entered into a database. This database happened to be Sequence Match. And after entering, the top 10 hits were listed and uploaded. As you can see, every single hit that was uploaded for Sequence Match happened to be the pathogen that we were assigned in meningitis. They all have an S_AB score of 0.995, except the very last one that has a score of 0.991. These numbers in the last column, accession numbers, are just numbers that help identify the pathogen in different databases. Along with Sequence Match, we were given hierarchical results. We were also given Bayesian classifier results. And we were asked to compare these two, Sequence Match versus Bayesian classifier. Upon comparison, we noticed that the results obtained were the same. Sequence Match and Bayesian classifier both have domain bacteria, phylum Proteobacteria, class Beta Proteobacteria, order Neisseria, family Neisseriaceae, and genus Neisseria. Although they were both the same, these results might have differed because these are two different programs trying to classify DNA sequences. Next, we have the results from the BLAST DNA sequence analysis. Unlike the Sequence Match DNA sequence analysis, the BLAST results showed 10 different species. However, the most important one is the number one hit, which happens to be in meningitis. However, this is number one because it had the max score of 2,492, and the other matches below had lower scores. Here is our phylogenetic tree horizontal. This shows the many different species that were listed in the previous DNA match hit from BLAST. Now, all of these hits did have the genus Neisseria, they just did not have the species meningitis. And these different species are listed here in a horizontal form. And next, we have our phylogenetic tree in a slanted version, which just shows a different direction and a different perspective of the genetic trees. All right, now moving along to talking about the actual pathogen in meningitis. We have pathogenesis, which is how a disease develops. So first off, in meningitis is often referred to as meningococcus. Coccus meaning the morphology, which is the round balls. It is exclusively a human pathogen, which means that it is only found in humans. It can't be found in any other organisms like animals, insects, etc. This pathogen also occurs worldwide in both epidemic and endemic forms, including countries such as Africa and Asia. This pathogen is spread through saliva and respiratory secretions in different things such as coughing, kissing, sneezing, and even sharing sources of fresh water. And lastly, this pathogen is found in the nasopharynx and it often lives there as bacteria. Next, we have the virulence. which is how detrimental a disease might be. So, N. meningitis has lipopolysaccharides found on its outer membrane. It's also known as an endotoxin. This pathogen is responsible for septic shock and hemorrhage due to the destruction of red blood cells. A few other virulence factors include the polysaccharide capsule, which prevents the host phagocytosis. Polysaccharide capsule also helps aid in invasion of the host immune response. And lastly, this pathogen infects the cell by attaching to it with pili and also with other proteins called OPA and OPC. Disease. This talks about the different diseases that can arise from N. meningitis. So one of the most common diseases that can arise is meningitis. And it can also cause other meningococcal diseases, including the life-threatening meningococcemia. Symptoms of the bacterial meningitis include fatigue, purpura-like rash, which is shown here in this image, fever, headache, coma, and in the most extreme cases, death. And treatments for this disease include hospitalization with antibiotics. Epidemiology. N. meningitis is responsible for epidemics in Africa and Asia. And as you can see in this green belt, this is known as the meningitis belt. It is a major cause of illness, development impairment, and death in childhood. Every year, 2,500 to 3,500 people are infected in the U.S., and children younger than age 5 are at the greatest risk. History. N. meningitis was first observed in 1884 by scientists Ettore Marchifava and Angelo Celli, here and here. It was found in the cerebral spinal fluid cells. However, in 1887, Anton Weisselbaum isolated the bacterium from cerebral spinal fluid cells of patients that had bacterial meningitis. Lastly, we have some fun facts about N. meningitis. About 10% of adults are carriers of the disease in their nasopharynx. It is also one of the leading causes of bacterial meningitis. N. meningitis can be spread during coughing, sneezing, kissing, chewing on toys, and sharing sources of fresh water. The fatality risk of this disease is 15% within 12 hours of infection, which means that out of all the people that end up getting this infection, 15% of the people will die, only within the first 12 hours of infection. And lastly, the first vaccine was licensed in 2005, but vaccine development is still occurring even today. Now class, can anybody tell me what they've learned today? You. Well, I learned that N. meningitis was one of the leading causes for bacterial meningitis. Bacterial meningitis has the greatest risk in children younger than 5, and 10% of all adults have the bacteria living in their nasopharynx. Great answer, Jessica. Now I hope everyone enjoyed the lesson today on N. meningitis, and I hope you all have a wonderful day. See you tomorrow. Bye.